Hello, everyone. Welcome to the MIF++ seminar. It's a great pleasure to introduce Dr. Olga Anosova, who will talk about duplicates and high-profile structural databases. Olga, please, over to you.
Okay. Thank you very much, Vitaly. And well, let's try to. Okay. So this is an unusual session because firstly, it's a joint session with MIF++ and CDT doctoral training in materials chemistry. And secondly, because unusually for this kind of a seminar, we will not have much mathematical formulas today as well. Then we'll have, I think, just one definition, but a pretty important one. Oh, but I wanted to start with some connection with the previous stuff that Vitaly demonstrated, and demonstrated that, yes, unfortunately, those kind of difficulties have not disappeared yet. So yeah, this is what, thank you, I am fine for now. So this is what AI now tries to do with labeling, sort of paying the same to the humans who did not label things properly enough. And yes, that's one example, that's another example. Which means that, we used to think that it's just confusing for AI training, but actually we are now feeding the consequences of whatever imprecise labeling stuff and so on we presented ourselves, we introduced. That's why it is important to indeed see what we need to make the things more structured, more defined, and more useful. And also today, mostly will see the consequences of what happens if we don't do that. In particular, we'll consider the cases when we consider the exact duplicates in famous databases. Let me notice that the quick discovery of those new duplicates is now possible with well-developed geometric theory behind it. So let's start with the data that probably you deal with in some sense, one way or another. So yeah, that's how we often represent this data, the crystal. Can you recognize the crystal on the left? Any ideas? Ice, well done. So you can notice one large atom, two smaller atoms, and a nicely ordered collection of them, which means that most probably it will be ice, and indeed it is. It is so-called hexagonal form of ice. Well, any idea what's on the right? Yeah, that's a more complicated one, and you maybe haven't dealt with that yet. That's representative from a metal organic framework, so it's something more complicated. But that's how we see things, or try to represent them for ourselves, because we try to be sort of visual creatures. How are those things represented in digital forms? So from images now to crystals. And we've introduced the definition of a crystal, or more precisely periodic crystal, as a periodic point set. So firstly, we need, well, that's everything, we need some sort of origin to start our representation from. In mathematical terms, it's called basis, more precisely linear basis. And then any linear basis will define a unit cell. So here in the picture, we have two vector bases, V1 and V2, typical Cartesian vectors. Yeah, and then the unit cell is just something that it can be sort of spun or put on those vectors. Then those vectors, if we repeat the unit cell, define a lattice, again, are mathematical terms representing what a lattice is. But that's not the only thing, because sometimes we can have some atoms in between within those unit cells, within the lattice, and that we can call a motif of atoms, And then the periodic crystal, it is just the combination of a unit cell that defines the coordinate system, a lattice, and a motif within this lattice. So why is it still non-trivial? Well, you can see several blobs of color, green, blue, red. You can probably guess that those are all unit cells, just spanned by different vectors. Yeah. So, and that's one of the problems. Another problem is here. Suppose we decided that we are just dealing with square lattices or anything that resembles square lattices. Great. Now we add some motif. Yeah. So suppose we added motif like that for blue atoms, for red atoms. Okay. And we are just dealing with square lattices. But now let's see this square unit cell and this square motif, yeah, well, for blue, for red. And now this motif within again square unit cell, this one. So formally, motif looks differently, yeah? Different combination of reds and blues. Are those same crystals or not? Well, actually, the solution is in front of you. That's why we know that it's the same. Yeah, we can repeat that motif using our lattice many times and see that it's just different cutouts of exactly the same crystal structure. But yeah, that's just one simple example. So yeah, and they look quite different to motifs within unit cells. And you can imagine that the things become even more complicated if at least one of the atoms slightly vibrates and shifts its position. Then, of course, if you have more atoms within the motif, things become greatly complicated. And then the question, what we call same or different, is not trivial anymore. And it's not trivial not only for us who try to look at that, but also for computers who presumably should be quite good at computations of different bases and different atoms motifs and so on. So, well, does it happen in real life or not real life data? Here is an example from a one of the crystallographic information files, CIF files, who did deal with this kind of files already. Quite a few people, but not everybody. So that's one of the typical way to represent crystal structures. Indeed, it has a unit cell parameters as 3 lengths and 3 angles, and then atoms or motif is represented within this unit cell as so-called fractional coordinates, and those are fractional because, yes, it's fractional of the unit cell, and to get the full structure, you will need to repeat that into Euclidean coordinates again and again. Well, is everything totally perfect in this kind of files? Well, you can notice that angle here. And that's an example of how teeny tiny perturbation really happen in real life measurements. Yeah, because it should have been in the ideal world maybe 60 degrees, but it's not. Because especially if it's a real life data, then teeny tiny perturbations will happen and will be measured, whether it or not, or if you wish it or not. So how does it impact whatever happens? So this is the first story that I wanted to tell you. And that is a story about Google GNoME, as they call it, but I like to call it GNoME. You probably heard about AlphaFold. Yeah, so AlphaFold got Nobel Prize for solving protein folding problem. And Google really wanted to repeat the same success with materials. So they've issued the new AI called GNoME or GNoME. and lots of new crystals. And as they say on the website, that's AI, this new crystals represent 800 years worth of human knowledge. Beautiful, isn't it? And the whole idea was even better because they decided to go further now and actually test in practice the crystals that were created. And for that, they've partnered with Berkeley A-Lab. And that is automated laboratory that, well, tries to automatically create the materials or synthesize the materials from some files. So what was the result? The result was that they've published a separate paper and claimed to synthesize 43 out of 58 materials. Okay, so that sounds like a perfect fairy tale, right? Well, unfortunately, it was a fairy tale because pretty soon chemists were looking at the materials. And that was one of the first reviews about the synthesized materials. And it claimed that basically none of the materials were new and the large majority were misclassified. And there was probably a small number that was correctly identified. But already know. Well, this is sort of an interesting again story in itself, because actually there are, I think, 3 materials which probably were not in the training data as chemists thought. They were discovered and synthesized a bit later, but everybody thought that they were not in the training data, which means that, okay, 3 out of 800 years of knowledge is probably not that bad. Unfortunately, it again was a fairy tale, because the paper by Dan Vidalson and Vitali, recently published, demonstrated that essentially this 3 is unfortunately equal to 0, because all crystals had well previously known duplicates in their one-dimensional databases, IUCrJ databases, and while all of them were not new. That was not just noticed because people didn't have the tools to properly compare the crystals. And one of the issues why it was happening, well, a simple demonstration on the previous page. Because those motifs, those crystals looked different, not only to the human eye, but if you try to compare typical simple methods, try to compare them using simple methods. Okay, so that was about synthesized materials, but we still have something like 2 million, okay, predictions. Actually, GNoME publicized just 384K stable crystals. Well, there's some description, very short one, what they mean by stable. So it was close to the, well, how it's typically done, close to the boundary of the energetic convex skull. So presumably those materials should have been stable. But the chemists again started to look at the predictions, and there was the next review now about Google GNoME dataset itself. And whatever they looked at, they found some scant evidence of, well, anything that is novel or credible or useful, because some of them were just very unstable and containing radioactive materials. They tried to do it by hand, but which is a bit hard with this number of elements or predictions. So we tried to do it in a slightly more systematic way and published some of the outcomes in a couple of papers. The first one actually introduced the exact definition of periodic crystals, which actually allowed us to do this kind of quick performances, quick comparisons. So what we've discovered that unfortunately there are thousands of new duplicates, and many of the predictions are just, again, not novel. They're very close, small perturbations from whatever was already known. And since, well, smallest interatomic distance is 1 angstrom, and here the distances are much smaller, that's indeed just the perturbations, not some kind of reasonable adjustment of the material. But that was, again, just the beginning of the story when we saw, and here is a nice example. Yeah, well, I can ask you, do you think that those crystals are same or different? Probably you already can guess the correct label or answer from how I'm presenting things and where I'm leading. Well, believe it or not, indeed, the coordinates, the geometric form of all of them is presumably the same. So with the middle one and the right one, that is particularly, well, sort of believable. Yeah, you can sort of turn them and then put them against each other, and they're believably similar. So it's still unbelievable because atoms should be different, yeah, and quite strikingly different, which means that having same geometric structure with same geometric, well, anyway, how we represent it, well, that's quite improbable, with having so different composition, but exactly same geometry. Well, how about the first one? Does it look similar to the second or third? Well, that's AI creation. Yeah, it's clever, it's brilliant, it's very creative. And it doesn't even look like anything from a sort of more or less humanly created in the ICSD materials project. Yeah, this one, in fact, if you look at it properly, it is again represents exactly the same geometric structure. Well, composition again is different. Is it possible? Well, it is doubtful. Any questions here? Yeah. So that's just to demonstrate the difficulty in comparisons of crystallographic structures, and that indeed the exact definition, good quality definition, and good quality methods of comparison are absolutely essential. Even we, with our human brains and eyes, can sometimes not detect. But yeah, the story of GNoME is even more interesting because we didn't even need to see at more complicated algorithms or more complicated methods of comparison. It turns out to be sufficient, largely sufficient in many cases, just to look at the files, C files that I presented before, which means, well, unit cell parameters and fractional coordinates. And if we compare them, even then, we have some interesting finds. So firstly, some of the CIFs were just identical texts. I don't know. Secondly, if we, we do not compare the tests, we just compare the numbers, well, the number of coincidences grows. If we start rounding, because essentially, well, we can think of rounding as of going back to the natural perturbations, then the number of coincidences grow any further. And well, let me notice that there is a group of 9 CIF files, so 9 materials that coincide numerically but not as texts. Yeah, 9 of them. We'll see them in a moment. Well, if we compare not fractional coordinates, because I've mentioned that yes, we probably need to see the whole structure, not just the cutout of it, not just fractional coordinates, then the numbers are slightly different, well, in both directions. But if you round it to 1 digit, which we might think is more realistic sort of estimations, then all the numbers of coincidences, wow, they're strong. Fake it till you make it, right? Yeah, well, okay. They seem to try really hard. Well, let's have a look at the largest group of 9 duplicates. And you can see that though the geometry, as I mentioned, is indeed the same, those are the names, you're welcome to look in the files and see it by yourself. All the parameters, unit cells, and the coordinates are the same. The chemical formulas are quite strikingly different. And you can see that probably some kind of interesting optimization indeed was happening, but is that a clever kind of optimization? I don't know. By the way, you can maybe count how many atoms are you dealing with. Yeah, I think so. 28. Yeah, 28 for those ones. But the most frequent number of atoms in the whole data set, with approximately 50,000 predictions, accounting for this number was 36. And you can sort of guess why this number. That is a very nice number that is combined from many other whole numbers divided by many other parameters. So it's easy to optimize those sort of atomic structure, atomic composition if one really is determined to do that. Well, another interesting example is this one. It is claimed that we have four nitrogens in here. But if you look at the picture, yeah, how many atoms can we see? Well, those are only two. They're slightly smaller than the rest, not the four. Yeah, all of the others are different. So what's going on here? Well, it turns out that indeed one of the nitrogens is just repeated twice, but it is given a different number. And
it's not nitrogen, it's sodium.
Sorry? Oh, Na, sodium. Yeah. Thank you. Thank you very much. Yeah, I'm not a chemist at all. But that thing, even I can notice. The numbers I can notice, the names probably not. Well, I really think that 2,800,000 years of knowledge, it's not much. Why couldn't they create more than just 2 million predictions? Yeah, doing something like that. So I had this idea: can we create on the cheap, really easily, can we beat Google GNoME? So the idea is we take any structure and we apply the same idea as it seems to apply, that is, apply small perturbation. So you can see on the left is one structure. I don't remember which one it was, but I do remember sodium. Yeah. Yeah, and the second one, believe it or not, it's the same, just with small perturbations applied to the unit cell. It is slightly skewed as well as the position on that. And indeed, well, to our eye, it looks quite different. How many of those we can create? Well, actually, it was a school project of my daughter, a final year school project. She created indeed a tool that takes a CIF file, applies slight perturbations, and create absolutely novel prediction, symbolizing how many years of human knowledge you wish. And it was possible, obviously, to create these kind of 2 million realistic crystals in just 16 hours on a small laptop using the knowledge of a school child. Well, good. So, well, still something to work on. If large AI and large AI teams would like to beat a school child. But actually, is it just unique to AI, these kind of issues? Well, probably you can guess again, but probably not. Because AI just repeats what we did before and just do it quicker and in larger amounts. And I could have demonstrated some duplicates on the crystallographic databases, but I decided to go in a slightly different direction. Because, well, next to crystallography, people quite often mention structural biology. Because proteins also are experimentally measured by similar methods, well, also X-ray structures, and then, well, quite a lot of what's going on with proteins is quite similar to what's going on with crystals. And even, for example, on a crystallographic meeting, crystallographic congresses, well, at least third is typically devoted to proteins and protein structures now. So, what is the most famous experimental data with proteins? So, with crystals, there could be several databases mentioned. That's why probably it's a bit hard to notice and trace the duplicates of what's going on in crystallography. In proteins, it's a bit easier because there is a unique protein data bank that indeed is a collection of, I think, really all known experimental proteins. So each experimentally derived or experimentally measured protein is put in also C4, more precisely, MMC file. Each file is decoded by a 4-symbol code, and 4 letters are still enough, which means that, yeah, not too many of them. And, well, those are some representatives of those proteins within the files. Do you think those are same or different? Yeah, very good answer. And the answer will be, I don't know, right? Because from this representation, you can't say anything. And indeed, well, the best answer, it's better to say that I don't know, something that give answers that might be wrong, as Richard Feynman famously said. So, indeed, those are quite large proteins. And within each protein, which is not visible from here, we can have several molecules or entities, and each consists of several chains, and also there can be several versions of them in the same file, all in the same sort of, all in the same file in the same place. Which means that comparing them is not very easy. So what we did, we started from the simple thing, the protein backbone. And the protein backbone is the heart of every protein chain. Because proteins, in fact, are just large biomolecules that are formed by 20 amino acids. Well, approximately 20 typical amino acids. And those amino acids, so you've seen in molecules, they form, well, in crystals, they can combine in very complicated periodic structures. In proteins, it's simpler in a sense that those amino acids are connected sequentially from one amino acid to another by so-called peptide bonds. And another same thing for each amino acid is the existence of nitrogen, alpha carbon, and carbonyl carbon. And, well, there are several definitions of a protein backbone, but we'll say that protein backbone, that's just a connection of nitrogen, alpha carbon, and carbonyl carbon. So our idea was, okay, to take just those protein backbones, and we'll start by comparing those backbones of those covalent structures and see how things go. We were able to indeed apply a similar approach as Vitaly and his team applied to crystals. And yeah, we found that in this database of experimental structures, there also do exist exact geometric duplicates. Most interestingly, well, approximately 700 pairs are in different PDB entries, which means that, yeah, they are coming from different experiments, different structures. The question is, how could it be if those were measured experimentally? And, well, we can sort of imagine that proteins are quite flexible creatures, and they do not like to be in exactly the same geometric shape or form. So we started looking inside each of the cases even closer. For example, there were 9 pairs with different primary sequences of amino acids, which means that amino acids are different, but the coordinates are still the same. How could it be? So that is the first collection of 9 different amino acids, 2 chains, quite long ones, and 9 different amino acids. And the author actually accepted the duplication of them, saying that, well, PDB entries can multiply on their own, and so on and so on. Okay. How about other ones? Well, there are examples where just one amino acid is different, glycine replaced by gluten. Well, okay. Those examples are quite
It's glutamine,
interesting.
not glycine.
Sorry?
It is glutamine, not glycine.
Yeah. So look at the last pairs of structures. They have quite a bit of different amino acids. So what's going on in there? So let's try to look at them. So the first set of files, they are coming from a similar type of viruses, whilst the second file is coming from a different type of viruses. Okay, it's still viruses, but the thing is that they are known for quite different mechanisms and quite different interactions with the enzymes and interactions with proteins and with receptors, and different disease results, different diseases or different disease outcomes as the result of that. Which means if they indeed have the same structure, that sort of makes it questionable the whole theory of the lock mechanism, yeah, that the shape defines interaction with everything around it. Then what actually are we developing? Where should we go? Should we reconsider all the theories? Well, those are picturesque representations of 2 of them. Do you think they're the same or different? Yeah, they look the same now, and indeed they are the same even when applying coordinate by coordinate comparison. Well, there are 2, not 3 of them. Why? Well, because actually we released part of our results to the PDB team, and one of the mentioned file, this one here, was quickly quietly updated, and now it's slightly different. 2 from different viruses are still the same, which means that, well, yeah, something's going on there. Okay, something's going on there, but well, we are not biologists. We can't really analyze what's going on. I can't even remember the names of the atoms. So we've teamed with biologists. Here is another example, which was sort of easy to see. Again, one of the amino acids are swapped by another. This one, yeah, you can just see that everything is absolutely the same except of this swap. So we've teamed with biologists, Alex Wadera and his team, who really looked at least some of the examples. And no, they were not able to look in precisely 700 plus or even more than they found. So they've looked at the filtered out best possible results with the best resolution of 4 angstroms or better. And well, as a result, produced a table of manually checked instances. Here is a sample of this table. So the first PDB, the second PDB, the number of residues and the number of different amino acids in them, and the quality of the results. Most probably they should be good. And here is the full table of 553, I think, instances. They're analyzed. The symptomatic file has way more. Well, and yes, firstly, indeed, they were duplicates. Secondly, let me notice that the methods that we do apply, they do not allow false positive, false negative, something like that. So that is totally justified geometrical methods, which means that if you say that they coincide, they do coincide. Hence, the main reason for biologists to look at them was actually trying to see what's going on, what was the reason, and how to avoid it, or how to make life better and database better. So, well, one of the case was the first one, here with 9 different amino acids confirmed by the author, subsequent redeposition, but still in the PDB. And I think they've noticed it some years ago. Everybody knows about that, or many people know about that, but it's still there, still unresolved. Some of them were deposited very close together. Maybe there was a mistake in one of the depositions, maybe some uncertainty, but they still are both there. Sometimes the depositions should be different because they have quite different descriptions, but still the geometric structure is the same. Well, for example, the first case hints at possibly non-experimental modeling, when people thought that something should look similar to another protein, and then just substituted the numbers from the first protein into the second guessed protein. Well, can we call it experimental structure? I don't know. Well, and some of them were sort of, well, you see, it's diffraction patterns, and then if the picture is not perfect, it can sometimes be refined in different ways. For example, by removing some of the atoms so that the suggested protein fits into everything a bit better. Well, can we say that it's a different structure? Maybe one of them is a different and real structure, but having 2 of them in the database, well, it's questionable. And most of them, unfortunately, fall into the quite a substantial cluster of other peculiarities, which means that it's even hard to see what can be done in that direction to avoid those cases. And actually, we can look at other measurements, especially after we found many duplicates using our methods. It is possible to look at other possible measurements by RMSD, and there are even more of those, not being exact duplicates, but being quite close to each other, which means that, yeah, maybe some of them are expectedly so, but some probably not, especially if we have different sequences. Well, for identical sequences, it's also quite interesting why they coincide so well for so flexible structures. Maybe that's correct, but some explorations might be needed. Well, why is it important? Because, well, you remember this file, right? And it's a famous GNoME principle. Do you know what this abbreviation means? Quite famous in computer science. GNoME principle. Garbage in, garbage out. Which means that for GNoME, for genome, really depends on the data being fed, on the methods that it was fed, on the algorithms, and on the evaluation that we offer. Yeah? Which means that if we want anything good, we need input nice stuff, and also have very well-defined methods of at least evaluating that stuff. And well, that's what the data science training really wants. To building this basis knowledge to allow us to build our own understanding of what's going on. Yeah? And it's sort of feel for junior generation, because now whatever we do, whatever data we produce, this data first will be collected, then it will be saved, then it will be used. And that's quite a unique situation which I think none of your teachers, including me, ever had. We all had situations when our mistakes were not recorded and were forgotten. Now it's not the case. Okay? And while we are having an increasing amount of tools that help us produce, but there's one thing that is not changed and probably will not shift from us in the foreseeable future. And that is responsibility. Because whatever program we make, it's all always the claim that, well, a user is using it at their own dangers. Yeah, should check everything. It's your own responsibility. So yeah, production speed changes, but responsibility is still on us. So that's why we need a really good basic understanding of evaluators and what they actually mean. And the good news is that here in this data science training and in your CDT, you have all opportunities to learn. Well, welcome to this CDT. That's all for today. Those are some links that I've mentioned. Any questions?
Thank you. Thank you. We will stop the recording. And
How to actually stop the recording?
So, Vitaly, I have a question.
yeah, of course, Moise.
Yes, so it was maybe because I couldn't hear everything well, maybe I haven't heard something, but when